Hello, a very good afternoon to everyone who have joined us today. Welcome to the webinar on the use of advanced molecular tools in infectious disease research. This event is organized by the Center for Biomedical and Nutrition Research in collaboration with the Center for Research on Communicable Diseases. Present among us today is also the chairperson of the Center for Biomedical and Nutrition Research, Dr. Eddie Chia Seong Guan, and the former and current chairpersons of Center for Research and Communicable Diseases, Prof. Miao and also Prof. Yap. I am Alicia, the MC and one of the moderators for today's event. Before we begin, let me brief all of you on some housekeeping rules. First, take note that your audio will be automatically muted for the entire session to avoid interrupting the speakers during their presentation. So to make sure that we are helping you as best as we can, we have a general chat box available for you. Feel free to drop your questions on the topic or share your struggles and experiences through the general chat box. The moderators will collect and relay all your questions to the speakers during the Q&A sessions at the end of their respective presentations. For your information, this webinar will be recorded. By continuing in this meeting, you are consenting to be recorded. And you don't have to worry about having to take down notes, as we will be sending the session's recording your way on a later date. However, do take note that only those who have filled in the sign-in form will receive the video recording. So make sure to sign in your attendance as soon as possible. All participants, you are reminded to fill in the sign-in form once you have joined and the feedback form at the end of the meeting. The links to these forms will be periodically shared in the general chat box. All Utah students, please be reminded that you are required to fill in the sign-out form as well in order to collect your USSDC points for attending this webinar. As always, there will be a virtual photography session for all participants at the end of this webinar. We hope that you can join us until the end of the event. Here is a quick run-through on the program schedule for today. The first talk will be by Associate Professor Dr. Chan Kok Gan, with the title of his talk, Next Generation Sequencing, Enabler of Next Generation Microbiology. Professor Chan's talk will be moderated by Dr. Chu Kwok Cheong. The second talk will be by Dr. Joshua Ang Sinter, who will be talking on Cas9-based gene drive for the genetic control of mosquito vectors. I will be the moderator for Dr. Joshua's talk. A short Q&A session will follow after every talk. We will end today's event with a group photo session at 5:30 p.m. Now, I would like to introduce to you the moderator for our first talk, Assistant Professor Dr. Chu Kwok Cheong. He graduated with a BSc Honours in Biochemistry from the University of Saskatchewan, Saskatoon, Canada. Subsequently, Dr. Chu obtained his MSc and PhD in Molecular Biology from University Science Malaysia, Penang, Malaysia. His research interest is in the molecular genetics of the nitrogen fixation, NIF genes, and the extended spectrum beta-lactamase, the SBL genes, from environmental samples. I shall now pass the time to Dr. Chu, who will be introducing our first speaker for the day. Over to you, Dr. Chu.
Okay, thank you, Dr. Alicia, for the kind introduction. Okay, a very good afternoon to our distinguished guests, committee members, and all participants. We are indeed honored to have with us Associate Professor Dr. Chan Kok Gan. And before we start, allow me to go through an impressive biography of our distinguished guests. Dr. Chan is a Commonwealth scholar trained in molecular microbiology with a rare combination of expertise in law and science. He is very prolific researcher who has published more than 451 articles, authored several books, authored several chapters as well as books. He regularly publishes in some of the world's top journals, which include Nature, Nature Communication, Molecular Ecology, just to name a few. So in a span of 12 years, his current age index is 59. He is well known for his work in bacterial quorum quenching and genome biology. He was recently named as one of the world's top 2% scientists for publication in microbiology by Stanford University. Dr. Chan was elected as the Fellow of the Academy of Science of Malaysia in 2017. He won many international awards, including Commonwealth Scholarship, Prime Minister's Initiative 2 Award, JSPS Young Scientist Awards of Japan, as well as more than 10 French government fellowships. Dr. Chan has been awarded the Malaysian Torrey Science Foundation Science and Technology Award, and he has served as the ad hoc technical expert group member for the Convention on Biological Diversity, UNEP on Biosafety of GMO, as well as currently a Genetic Modification Advisor Committee member for Malaysia's Ministry of Energy and Natural Resources. So without further delay, please give a warm welcome to our distinguished guest, Associate Professor Dr. Chan Kok Gan. Dr. Chan, please.
Thank you, Dr. Chu, for the kind introduction, and I thank the organizer for inviting me to give a talk on sharing some of my research here. Please allow me to share the screen. Okay, does this work at your site? Can you see this PowerPoint? I mean, does it appear on your site, this PowerPoint?
Yes, we can.
Okay, good. So then I will just move on. And good afternoon, everyone. First of all, once again, thank you very much for inviting me to share some of my work on microbiology and also with the use of NGS. And I thank the organizer for this event so that I can showcase some of the work here. Okay. And myself is from University of Malaya, and I see quite a lot of familiar names in the participants. To all my friends, good to see you here. Okay, and I have several professorships from those universities, both national and international. And today I would like to share with you some of the work that I have done, and the title is Next Generation Sequencing. So enabler of next generation microbiology. Okay, now, so just to a quick flashback of what we have learned when, if you are my contemporary, that you are in the 1970s. So then in the 90s, when I did my undergraduate, so this is some of the, I still remember these lecture notes given by my mentor, Dr. Tan Eng Lee. And then he showed us the Anthony van Leeuwenhoek inventions. Then of course, later when I study more books, then I found out that he's not the first one who invented the simple microscope. Then, but that will be the basis for my bacteriology way back in 1994, 95, not long ago, I would say. Okay. Then in 1977, a young bright chap from Cambridge, Sanger, so Fred Sanger, which passed away a couple of years ago, and his technology is actually the cornerstone technology for my PhD work, where I did a lot of sequencing of bacterial. But that time we don't have next generation sequencing. So I always love to start this. Some of you may have seen this before, because this always remind me of the good old days when I was a PhD student in the millennium, the new millennium year 2000. So I once was told by my supervisor during his time sequencing, if you can get about 15 to 40 base pair, you can publish almost for sure in nucleic acid research, which is a very good journal, okay, high impact paper. Then I couldn't believe my ears when I was in the year 1999, in year 2000, when I did my PhD. But then at that time, I used the same technology from Fred Sanger, the dideoxy chain termination chemistry, to do the sequencing of my bacterial work. At that time, if you can get about 600 to 650 base pair, you are extremely good. And my supervisor at the time, Prof. Ko Chonglek, didn't allow us to outsource. He would prefer the students to do the sequencing. So then, of course, we are very well trained in this. And I have seen this myself with the radioactive labeling. I handled some of it when I was a student, but now I don't think the younger generation will see this anymore, which is almost literally not possible to do radioactive work. Then my students recently told me that the 16S sequencing, when they outsource, so it usually come back with more than 1,000 base pairs. So that's how the technology improved over the years. And just to refresh your mind for those who are not aware of the course, when the molecular genetics started the Human Genome Project in what we call the end of the last millennia. So it takes the 4 continent scientists to join hands together worldwide, from Japan, China, all the way to Europe and USA. So multiple governments, the Hugo project started and ended in year 2000, approximately $3 trillion US dollars. Well, I was a banker, so I've been trained to make you aware how much is this money. So if you lost track of $3 trillion dollars, so that is about, I think, 15 trillion ringgit. Okay, it's enough to build one Heathrow Airport Terminal 5. For those of you who are from London, then you should know. Or if you're a big fan of aeroplane, that would be 10 Boeing 787 or Airbus 380. If you are big fans of shopping, that will be about 40 Valley Mega Malls in Kuala Lumpur. Or if you are very academics minded, so that's about 250 secondary school Malaysian standards, okay? Not take into account the 5% inflation rate in our country, okay? So that's one human genome at that time, okay? So that will, let me set the scene. I just want to tell you that the genome biology used to be a very expensive work, almost not reachable by ordinary scientists like you and me, but time has changed, okay? Allows me to impress upon you some of the work and some of the literature that I have found for you. The first thing will be the functional genomics that actually we call it the RNA-seq. So at times when we are very saturated with the genome sequencing in my lab, so I started to tell one of my PhD students that why not we do something very simple, but yet we have not done it before. That's the first RNA-seq that we have done. So we published in PeerJ at the time a Q1 paper. And then the idea is very simple. So we use the nosocomial pathogens, which is Pseudomonas aeruginosa PL01, a very well-studied genome bacteria. So its genome has been fully sequenced. And then we started to look at the RNA changes as we subject it to the elevated temperature. So it is very simple. Get to what we call water bath. One heated to 42 degrees Celsius, the other one remain as 37. The idea is to mimic the patient's conditions when the pathogens invaded into the human body. Forget about the macrophages and all the immunity that will come in much later, okay? Once they penetrate through the body, the first thing that they encounter is actually the differentials of the temperature. So from the cooler environment, then they move it to the constant 37 degrees Celsius, and then followed by a fever or febrile conditions in the patient. So what will happen to the RNA populations? So the simple idea, okay, then we extract the RNA, fix the time, we extract the RNA, and then we start to sequence it. So this statistics analysis shows that the triplicates cluster very well. So on the top of the panel, these are the control, which is 37 and then 42. So the data are clustering very well, okay? They do not jump up. That's good, okay? Then we look at some statistical value changes. And also we found that the control and the gene expressed at the control temperature and also elevated temperatures are quite different. So these are the table that I list down for you, which are what we call significantly expressed up and down regulated. So what do we get? Okay, we actually get a very good surprise when we look at this, what we call the table. So if you're familiar with the heat shock studies, okay, be it in the Drosophila or the what we call pathogens, so the chaperone is always the first one to express. So even without working with the details, when the postdoc shows me the data, so first thing that I look at, of course, is the chaperone. So true enough, in the chaperone, it is highly upregulated. So deep in my heart, then I know that these data are genuine from my students, okay, without putting the CCTV in the lab. Then when I go through the list, something quite surprising to me is that there are small regulatory RNA, which is very upregulated in the transcriptome or the RNA populations in the pathogens. So, and that has not been reported by the literatures before. So we then also found out that this small regulatory RNA in the upregulated table is actually a master regulon for the quorum sensing. And for those of you who are not familiar with quorum sensing, it is a cell communications that regulate a cascade of genes expression at the population density level. In English, okay? So the bacteria gene will be expressed synchronously when similar species are gathered together in a confined habitat, which they are physically close enough, okay? And then they express the genes at the same time, at the same place. So this gives what we call certain competitive advantage to the pathogens. For example, when Pseudomonas PA01 invaded the human body, so they start to replicate, and when they achieve enough numbers, they need to express, for example, the virulence determinants genes at the same time in the same inoculation sites of the human body, so that they can outnumber the overwhelming response from the host, which they try to countermeasure the host defense system by act in unison. So this is what they call the quorum sensing. Then this master regulon, which is upregulated when they are subjected to high temperature. So the pathogens are actually being shocked, and then they start to coordinate their gene activities together. So this is very impressive, and that without what we call much being questions by the reviewer, the paper was accepted. So, and then of course, we followed by qPCR and certain experiments to confirm the data that we have, okay? And certain genes are downregulated, and in this case, we actually found that what we call the movement or the swarming and the swimming of the bacteria are actually slowed down. Perhaps they are actually dying at the high temperature at the 42 degrees Celsius, expressing the genes that are showing the virulence determinants are more important for survival rather than, you know, swarming and swimming. So that will be the first data that I would like to share with you. And then we also work on the NGS, where we study the multi-drug resistant pathogens, and we also go for the plasmidome analysis that we work together. And the first examples that I can share with you is the Coliseum, which is the MCR1 work. We actually published this in the journal with one of the colleagues from Utah as well, Prof. Miao Yun Fong, from the Faculty of Medicine and Health Sciences in Sungai Long campus. Okay, so we get this E. coli, and then we actually did a quick antibiogram assay. So we found that it looks like they are what we call MCR1, Coliseum resistant. Then without knowing the genetic determinants, we start immediately to do the complete genome sequencing. And that time we have the PacBio. So not only we get the draft genome, but we can actually close the gaps. So all the genes will be disclosed at the molecular level and sort of readme like an open book. And then we quickly found that the MCR1 is actually found in the E. coli. We also did an extensive search among the E. coli that we found in the environment, in the farms. So we work with the farmers, we get the diagnostic lab to work with them. So we collect all this, and we know that colisteins are sometimes being used in the veterinary clinical setting as well. So, and then true enough, our predictions, we found quite a number of them. So then we quickly published that. There is a story to share with you on this, okay? These two papers, we're supposed actually to make it into Lancet, but we being careless, so we deposited the genome into the NCBI. So, and without knowing that the competitors in China, Nanjing, is actually, they were working on the same area as well. And of course, then they worked much faster than us. So they found the MCR1 gene that make it into the Lancet. So, and then they actually pinpoint to our NCBI database our strains collections that have this, okay? And this is where I get a very upset moment with my former PhD students, okay? So moral of the story, if you are the students who work with this kind of project, you should be a bit more sensitive when you work with the supervisors. So certain projects, when you want to release the data into the public domain, particularly the NCBI, so make sure that your data are ready for publications, okay? Now, so the colistin-resistant E. coli, what is it so mysterious is that in that particular work that we have worked together, and this is the work that we have sequenced the entire genome to complete. So we actually found that previously this genome actually caused a lot of confusions to the bioinformaticians with my collaborators, even in Australia. So we actually found out that once we complete the genome, then everything will be, like an open book. So we actually found that it's actually the plasmid that is actually causing a lot of problems, because they do behave like the chromosome. They have the origin of replication, same as a chromosome. So that actually caused a lot of confusions in the downstream analysis. But with the complete genome, then things are in place. So then we can actually look at the genetic determinants one by one, and not only the last fit defense drugs, colistin, that has been resistant. So this bacteria is also actually resistant to quite a number of other antimicrobial drugs agents, okay? And we found quite what we call a number of genes which has been in repetitive form. So we have multiple copies of this genetic, we call it pathogenomics islands, so that actually arranged in tandem in the genome. So, and there are actually a cascade of genes that sandwich in between the chromosome of these clinical isolates. So that results in a very complicated, what we call, this is where the colistin is embedded, okay? So this island is actually full of, it seems like the E. coli, this particular E. coli is actually very tidy, So they put all the things into one drawer or a genetic locus. So that actually quite surprising for us. And then we start to map this in a very clear form, okay? And not only they are chromosomal bond, like what the Nanjing has reported in Lancet, we also found that this is worrying, that they are actually found in the plasmid as well. And this is a multi-plasmid organisms. And you know that E. coli are very promiscuous in sharing the genetic materials among the members of the Enterobacteriaceae. So that actually explains why when we start to screen the farm pathogens, the E. coli, we start to see quite a lot of these colistin-resistant E. coli in the environment as well as in the human pathogens or clinical settings, okay? Then we start to solve the mysteries that we actually can arrange this according to their phylogenomic relationships, okay? So this is where they form a cluster here, and then they form a cluster here. What is confusing or causing the mystery is actually the multiple plasmid. That is actually very disturbing for those of you who have worked with plasmid before in the genome analysis, okay? But with the PacBio, the third generation NGS, things like this are actually quite simple to solve. Tricks to share with you, until today, I still learn the lesson that my former supervisor has told me, if possible, try not to outsource. But of course, that on the first conditions that you have the machines. So we have the privilege that we own. We have the machines in the UMHIR. So we have the PacBio, and instead of outsourcing it like some other groups did, so I told my students that I think you should do the NGS yourself, including the RA and the master students. So all the lab students from my lab are actually trained in NGS. Then the moral of the story here is that if you were to outsource this, even with the same platform that you use in PacBio, currently now you have the nanopore, you will still miss the plasmid. Allows me to share with you the tricks that we have done. This has been actually, we are lucky to be advised by the designer of the machines, Steve Turner, who actually told us that if you follow the protocols, most likely you will miss it. Okay, he invented the PacBio, and also we have a Nobel Prize winner on board as my collaborator. He told us that if you do the size exclusion when you select the DNA, when you prepare the genomic library, when you have a very stringent, what we call filtering, that you actually filter out those small fragment of DNA, then most likely the small plasmid will then be filtered out. for example, when you do a gel elutions. So then the tricks that we do is that we almost gather every small fragments, although PacBio are meant for long read sequences, okay? But we, like the Malay Bahasa says that we sapu everything and we put it into the PacBio. So from there, we discovered that there are multiple plasmid that is actually present in this E. coli genomes, and we confirm it by the southern plotting as well, that we locate them. Okay, so another work that we have done together with the Australian group is that this is the E. coli VR50 that causes the asymptomatic bacteriuria. So the woman, the patients has been suffering from this bacteriuria repetitively, high recurrence. So, and again, this is quite mysterious to handle in the beginning. And you can see that the problem is actually this complex genetics makeup are actually confusing at the bioinformatics level. Again, when I took over the genetic materials, the DNA form from my collaborator in Australia, Queensland, so we quickly jump into PacBio and get the complete genome. Okay, so the first thing that is very appealing to us is again the genomic island, which is the GI-VR50. So the map is actually shown here. Okay, and this time we get multiple plasmid, yes, as usual. That looks like the R plasmid. Not only then, they carry the prophages that seems to be able to transmit, because these are complete prophages, they are not the quasi-prophage, okay? So, and that actually causing a lot of problems when you want to analyze it bioinformatically. So once you get the complete map, that's possible to analyze, okay? Then we start to look at the genetic determinants that cause this ER50, the asymptomatic bacteriuria in the human patients, okay? So my collaborator did this, what we call pathogenicity studies, okay? So this particular E. coli VR50 fails to trigger the interleukin-6, okay? But the alpha gene, which is able to colonize, and then we use the mouse bladder model to study alpha gene functions, okay? So what is interesting is this plasmid that I call the PVR50A. True enough, this is a conjugative plasmid. Yes, again, this is worrisome because they are able to conjugate and transfer by conjugations, and they carry the multi-drug resistant determinants here, okay? So that actually carry the multi-drug resistant in this particular plasmid that is actually able to share among all the members of Enterobacteriaceae. So this is the studies that actually confirm our finding. So that actually resulted in a very good publications that we actually worked together. And we realized that the VR50 is supposedly to be a commensal strain in the human body, which is very similar genomically, the makeup is very similar to the commensal E. coli K12, okay? But then they can persist in the human urinary tract and causing the asymptomatic bacteriuria simply by the gene acquisitions. First by the prophages, and then second by the R plasmid that carry the multi-drug resistant determinants that actually transmit easily across the E. coli kingdom. Okay, then I would like to share with you some of the work that we have done, but now no longer the functional, what we call genomics or the RNA-seq, or commonly known as transcriptome. Then we also work on the chipset called the chromatin immunoprecipitations, NGS. This is the one that I work with my alma mater, my former PhD supervisor, Paul Williams. Stefan Heap was my postdoc at the time. So at one day, when I got the UK Prime Minister 2 award that allows me to work for 3 years in UK. So I send my MSc students to work there. And then later she went on and do a PhD in Nottingham, UK. So in that 3 years, I went back to UK to do my research. And one of the things that we actually done during my sabbatical in UK is this, which is the genome-wide mapping of the RNA targets of the Pseudomonas aeruginosa, the same PAO1 that I got from Paul Williams to do my heat shock, if you remember the previous slides. Okay, but this time we do the RSM, which is a riboregulatory protein. So that actually resulted in a very good publication in NAR. If you remember the Fred Sanger slide, the 1977 slides that I told you, if you were born that early, you just do a Sanger sequencing, get 40 nucleotides, you will be able to publish that in nucleic acid research. But currently, if you want to get a paper into NAR, it is a very tough journey to go through, okay? So this is the paper that we spent about 2 or 3 years to get it done. And it's almost spoiled my entire Illumina MySeq. We'll share with you why is that the case. So the project for this RSM is actually very simple. We overexpress, we hypothesize that the small regulatory RSM protein are actually controlling a cascade of genes in this very well studied Pseudomonas aeruginosa P01. For those of you who work in medical microbiology, you should know that they used to call it, this bug has been studied to death. There is nothing new to be discovered. Think twice. Lesson 3 to be learned, okay? The first lesson is to act fast before you deposit your data into the public domain. And then the second one is to not think that something is mysterious. It's just simply that you may have a better technology to uncover it. And then the third one, when people tell you that the bugs has been studied to death, you think twice. Well, at that time, I'm ready to take the risk. So we have some financial support from the Prime Minister Award. So we decided to do this. Then what we did is actually very simple. We hypothesized that RSMN actually will bind, the protein will bound to the certain genomic region that will be important to regulate the phenotypes of these pathogens, particularly the genetic determinants for the virulence factors. Okay, so with this hypothesis in mind, we overexpress the RSMN gene in the PL01 so that they are full of this protein, and then naturally, if they are binding to the gene chromosomal DNA, so this protein DNA interaction will take place in the PL01. So then we harvest the genomic DNA. So as we harvest, we also harvest those RSM protein bound DNA region together. So then we further precipitate it using the what we call immunoprecipitation that is against the RSMN protein. So from there, with the total populations of the DNA, we further isolate based on ChIP-seq chromatin immunoprecipitations, those who are bound to RSMN protein. Okay, so yes, we get a very minute amount of this particular RSM bound DNA. Then my supervisor told me, nah, Kokang, this is for you. Okay, so then we have to crack our head when we do the nanodrop and the bioanalyzer. We almost detect a pure water, but we know the DNA is there because there's very minor reading from there. It doesn't pass the QC fixed by Illumina. Again, the moral of the story still applies. If you outsource it, then the commercial service provider will tell you it doesn't pass the QC. Thank you for your patronage. We return the samples to you. Okay, and bracket, you may actually throw it away because it doesn't pass the QC. So normally when I need to outsource, I will tell the sales, okay, even if it fails the QC, please do it. I'm willing to bear the negative results consequence, okay? But with this personal mindset that I have, we don't have to do this. So I told the students who help to do this, which is one of the co-authors of this paper, we will go on and do it no matter how. We actually get a nanogram of DNA. So that is actually almost impossible to do. And then we try to pull the samples. We did a lot of things to try to increase the concentration of this RSM bound DNA. And then what we did is that we still didn't get enough of it, but we went on to start the machines. And we are lucky, so we get the sequencing ChIP-seq done. So when we look at the DNA sequencing, then this transcriptome data show us that approximately about 200 populations of mRNA has been identified. And true enough, the hypothesis, they are actually encoding proteins which are important for acute and chronic virulence. So then we start to uncover the regulatory role of the RSM. So one of the key roles that RSM actually regulate on top of current sensing that I just told you, okay, is that RSM is actually regulating biofilm. No one has thought of that before. Then we start to do, the confocal microscopy, typical mushroom structures, 3D structures. So compared to those deletion mutants, then we actually start to confirm based on the ChIP-seq and then followed by the observation studies. Now, no longer, the Anthony van Leeuwenhoek kind of microscope. We use the confocal 3D microscope. We both have it in UK and my lab as well. So then we actually confirm this. And then we also did a lot of mutagenesis that study that actually confirmed that the RSM actually control the pale exopolysaccharide biosynthesis, okay, together with these C-di-GMP levels, okay. Now, they also control the CRC-Z, which is something new as well. and that has got something to do with the metabolism of these pathogens. Okay, so among this, then we start to look at more details. So we further confirm this activity by RSM that control the FHA1 genes, which is actually targeting the T type 6 secretion system in the PLO1, which is one of the key virulence determinants in the PLO1 as well. And that is actually directly controlled by the RSM. Again, this is another surprise that we have with a very painful ChIP-seq that we have done. Okay, so then we confirm it. They also interact with other RSM, which is called the RSMA. Okay, so then we do a shift, mobility gel shift, the band shifts to prove that they are actually interacting. And then together with this, they actually control the biofilm formations, such as those actually formed in the cystic fibrosis patients. When these genetic defects patients, which has a very extra mucus secreted, they're actually very conducive environment for the Pseudomonas and the Pandoroa to cohabit, and then actually what we call causing the biofilm formation in the patient's lung. So these are actually confirmed in the RSM ChIP-seq as well. So then this is the zoom in image that I've just shown you before, the confocal image. So that actually shows that the ChIP-seq data are actually correct. When we look at the predicted genes, we fish out some of the genes determinants to confirm by further biochemical assays. So these are all done. Okay. Then we further move into something more interesting, if it is not more challenging, I would say. Okay, so we look at microbiome. I think I've shown this to some of you before, but I don't mind to share with you. I quite like this idea, which is coming from my grandmaster, Professor Yong Hoi Sen. At one day, he told me, Kokan, you have done quite a lot of this. Why not you do some NGS with an insect? Okay, so he started to give me something that I am very familiar. The weaver ant, which is very common in the country. My housing area is full of these ants, which is very aggressive, and they are biocontrolled because they are able to protect the fruit trees. Okay, but they are very limited microbiota informations in the guts. So I start to work with Professor Yong, and then we get a PhD on board under my supervisions. So within 2 or 3 years, we get this published. So we have a distinctive microbiome pattern to be seen in the weaver ant that we collected in the urban area, rural area, and the suburb area. So our hypothesis is that it may have geographical differences as well because of the food that they prey on. Okay, so but to our surprise, when we get the Illumina sequencing data to be done again by my PhD students, so all these weaver ant samples collected from 3 zones actually shows a very homogeneous microbiome pattern. Okay, so it doesn't matter where they live, what they eat, it seems to be similar across the samples, as this bar chart shows, okay, which are dominated by the Lactobacillus and also Acetobacterus. So these are the 2 predominant families that are actually found in the guts of these weaver ants that we collected from, again, the rural area, suburb area, and the urban area. Okay, so we start to question why only 2 groups of this. We never doubt our NGS is wrong, because we are quite good at that, and we have repeated the experiment as well. Okay, it is reproducible. So questions, we start to see that the culture independent methods by NGS actually point to us that only 2 families are actually dominating. The 2 mafia are actually dominating in the guts of these 3 samples obtained from different zones. Okay, now the question is, why is that the case? Okay, so we look at this, we do a literature search. Acetobacteria and Lactobacillus both have one thing in common: they are acetophilic. They like acidic guts environment, which is full of phosphoric acid in the guts. Okay, and they are known to tolerate various sugar-rich and acidic environments. Okay, such as those in the weaver ant guts where they prey on the what we call the honeydew. Okay. So, and these are actually found in many species of the insects as well. Okay, and the high formic acid that the weaver ants in their salivary glands, they actually pump out a lot of phosphoric acid for 2 purposes. We know that they use that for attack. That's why when they bite you, they inject the formic acid. It causes a lot of what we call inflammatory reactions to the human being. But without knowing, and then once we publish this paper, In Germany, they published another paper. So they did a very sophisticated anatomy work on this weaver ant. They found that this weaver ant actually actively pump the formic acid in their salivary gland, on one hand as a defense weapon, on the other hand, they ingest it into their guts. So it serves dual purpose, okay? So when they ingest it through enough, because it's formic acid, they will make their guts very acidic. And that explains very well this putting our paper and the Germany paper together. So they artificially or evolutionarily created a very acidic environment, and they just want these two. So the question is that, so the microbiome, we all know that nowadays, 10 years ago when we started it, so we know that there are very trace amount of culturable bacteria being able to be grown in the synthetic medium. Being a traditional microbiologist, I know this well. I've been trained to play with agar media, but then 99% of it are not possible to be cultured. So then why could this data be useful to us as a traditional microbiologist? Like what I showed you in the beginning, Anthony Wendy Van Hoek. So I am very old-fashioned, call me traditional-minded, whatever you call me. I told my PhD students, yes, I got the first complete genome in Malaysia. I started this NGS in one of the very few pioneers, but I still love to see the bacteria growing in my, nutrient agar. So can we cultivate this dominant species in the weaver ant by using the microbiome data that we generated from the NGS? Or is there any linkage between the NGS data and the culture medium? Yes, that looks like possible. So we look at the microbiome, and then we look at the dominant families, two of them. So we design based on these two mafia species that are dominating the guts of the insect. So we have medium A, B, and C. Consistently, the pH is actually buffered to very strong acidic, 3.5. Okay, we put in acidic in one of the plate in case they are fastidious enough that they have to feed on the acid, organic acid. Then we give them a lot of sugar. Okay, so because the literature shows that they love these carbohydrates, so we give them. Okay, and alcohol we give them very minimal amount, as they are. Doesn't matter they're heterofermenter or homofermenter. So traditional bacteriology told me that once they ferment the sugar, no matter how ethanol will still be formed. Okay, so we put it there. We try to cook the food according to the customer's taste. Okay, and this reward seems to be very overwhelming. We are able to get a whole bunch of bacteria growing on this synthetic media. Then we quickly do a RFLP just to make sure that they are different. Then we pump it into multi-TOF mass spectrometry to look for the small proteomics. And that gives you the ID identifications in less than 12 seconds per ID. So within minutes, we are able to find out quite a number of new species and known species in this synthetic media that we have designed based on the NGS data. And that's why I would love to share with you. So for those of you who are working in the medical school in Sungai Long or in Faculty of Medicine or Health Sciences, you may not want to start straight away with the human samples. Play with the insects because you do not need, medical clearance, which take a long time to get approval. Warm up your skills and train it up. And that's where I adore the wisdom of Professor Yong, who are actually guiding me on this project. Then we start to do a complete phylogenetic analysis. We found that these isolates are actually very new, as highlighted where I have boxed it in red box here. Okay, so when I look at this phylogenetic tree, immediately I think we found the new species. Looks like we are able to find the two major families of bacteria there. Yes, it is true enough. So we found this, okay, and we reported it as a new genus. We have another one. This is the member of the Acetobacteria. And then we also found another one, which is on the way. We will publish it very soon. We have just sent it out for review. And this is a third paper in the series of our work. Okay, now I am approaching the end. Just bear with me for a while. NGS and pandemic. This I would like to share with you some of the thoughts that, can we just use the NGS to do something else or more related to, whatever you say, okay, your insect, I still want to work with the human. I understand some biomedical scientists are so diehard that they have to do clinical science. Fine, okay, NGS and pandemic. So what is the role of this in the era of COVID-19 where everyone is talking about it? And in March, I was actually in China when the pandemic took over. In the ground zero, I was in Hangzhou at that time in January. So they started to have mysterious pneumonia in Wuhan in December 2019. And in that December, I was actually in China for my research. So I was leaving China in January the next year, which is 2020. Then after that, the border was sealed until today. Okay, so in March, our country has the first MCO or the city lockdown. And then I start to read a lot of reports. Rather than, everyone is complaining about the lockdown, I spend a lot of time reading, and that's something that I enjoy during the MCO, and cooking, of course. Then I start to think of it. One of the things that I realized, which is very mysterious, this infects the lungs, and yet people have diarrhea, and they shred up, quite a number of the patients shred up the virus actually in the feces, and the discharge that they vomit, they actually contain the active virus there. And I'm not a virologist, but I did virology for 2 years, but this is something mysterious. On one hand, it infects the lungs, that has got nothing to do with the gut. And of course, after that, we know that it's because of the ACE2 receptor, which is highly expressed in the intestine of the human being. Then I start to realize that because I work on the metagenome in the environment, and I once wanted to remove the coliform McConkie agar that some of you may be very familiar. I was so saturated with McConkie agar. Then one day I asked my students, why not we go to the marine water? I know Malaysian waters are full of coliforms, okay? Then particularly Penang, where else, isn't it? And Port Dickson. So I told them, when you go back to hometown in Butterworth, get no need a gallon, just a liter of water. We do an MGS, and then we get the metagenome. We fish out the coliform bacteria. We can even count how many of them without the McConkie agar. So we did that. And I was repeating this idea when the COVID-19 started to take place last year in March. Then I was asking this, can we do a sewage water metagenome and fish out from there, instead of the bacteria genome, now the virome, and particularly the SARS-CoV-2 virome. And true enough, I think Holland is the first one who did it. Then after that, I think Singapore recently has started to monitor the apartment sewage wastewater. And I think Malaysia, UTM has been doing that as well. I'm not that sure anyway. So I put up a proposal to look for funding. It was turned down, but some other researcher got it. But it's good as long as someone is doing it. Then true enough, the Holland published a report. So they are able to find this virus genome through the metagenome in the wastewater treatment. And I think that search should go beyond the wastewater treatment, but rather the public water bodies in the what we call residential area. So you can do the metagenome, you can also do the metatranscriptome using the NGS approach on the wastewater or the public water system as well, just to monitor. And that will actually be very useful for public health management. And I would like to share with you another paper that I found, which I also read with interest, the One Health solution, when I was a consultant for AMR NGS in Joint Research Center in Italy, ISPRA. So I was the consultant for NGS, antimicrobial resistant. So, and that time, the EU has been promoting the One Health solution using NGS. So they want to, in short, monitor everything, especially on the AMR, antimicrobial resistant, using the NGS techniques. So this is where the expert like us was telling the European Commission what to do with that, okay? So, and in short, this is a catch-all metagenomic surveillance to prevent or to identify perhaps the pandemic and the next pandemic. How could that be done? So this is the one that actually possible. And I've been talking quite a lot with Professor Yong, someone very intelligent to tell me. So my initial idea is to screen the water system where the patient strap the virus into the water system. And it doesn't matter it's a COVID-19 virus or other viruses. You do a virome on the water system. You can also do that on the soil system, depending on the transmission mode. You can also, and this is where Prof. Yong shared his intelligence with me, search the wildlife as well. And we know that on and off, we still get the bird flu or the pandemic influenza happenings here and then, okay? And we know that migratory birds are flying, crossing the boundaries from one country to another. So when you sequence all this, okay, from the livestock, which is veterinary setting in the farm, like what I did, environment, we did that before, vectors and wildlife, Prof. Yong have done that quite a lot. healthy humans and the patients. So perhaps the Utah School of Medicine and Health Sciences can start to think of it, and then monitor all this catch-all situation. You need to apply a big funding for that. Okay, I'm not sure whether MOSTI or the MOHI will be a good source of funding for this. Write up and form a multiple teams, perhaps a multiple institutions to go for one health organizations. Get someone who is very good in bioinformatics to look at different pathogens, not only virome, perhaps the bacterial microbiota diversity as well. Okay, so then the wildlife, you remember the lab leak stories and the zoonotic origin of the SARS-CoV-2 virus. On one hand, certain countries are promoting this theory. On the other, they are actually talking about lab leak, and natural evolutions. So you can just stop the quarreling by just monitoring the wildlife. If you can find it in the wildlife that has hardly interact with the human, and that actually solve the debates. And also you will be able to identify a lot of new viruses from there. Okay, now, so can the NGS be useful in the mutants evolution studies and also the vaccines design? Yes, why not? Okay, so these are the few papers that I actually gather and to prove that my concept is correct. So you can actually have a machine deep learning to predict what kind of mutants that will emerge. Okay, so now we know that the Omicron is on the way, and they suspect that it's actually originated from an HIV patient. And I have hypothesized that, although I didn't publish that, but it's easily predictable, isn't it? If you understand Charles Darwinism in a very simple context, the virus is there. In order to survive, they have to mutate. And they are RNA virus. What do you expect? They are not stable genome. And they're proofreading. A simple year one molecular biology will tell you RNA is not stable. RNA viruses are prone to error in replicating their genome. So somewhat pressurized by your immune system, be it weak, moderate, or strong, okay, you are driving the evolutions. Like it or not, the virus will evolve. And for those who are very scared of the mutants or the variants, you don't have to be scared. Embrace it, live with it. They will mutate, okay? So then in this Nature Microbiology, these groups of scientists from the United States, they start to predict it using the machine deep learning. They predicted 6 mutants. Unfortunately, the Omicron is not something that they have predicted. Just like a lottery striking, they miss, okay? But it will be good to know through the machines learning what kind of mutants are emerging. For those of you who are very good at, animal model, and if you're allowed to handle the live virus yourself, inject it into, HIV model animal, and then let it naturally evolve, mutate it, and then try to isolate the mutants. But this is a highly dangerous experiment to do. But why not? Okay? If you are interested in the very high-end R&D, trying to look for the mutation mapping. And that can only be done by NGS. Okay, you sequence the entire genome. It doesn't matter you are using in silico methods to predict the mutant, or you actually isolate from the field, or you actually isolate from the model, animal model. Okay, just keep sequencing it until you find the new mutants and see the serology studies whether it works or not. And then you get a crystal structures or the 3D structures. You can do a biomolecular docking to study the antimicrobial or antiviral drug discovery for some of you who are interested in pharmacology. So this can be done through the help of the NGS. Okay, so and then you can actually track them, like some of you who are interested to follow the emergence of variants. NGS is the only way, and that will actually have a direct impact on the public health policy. I think the South Africa has been very good model to discuss with you. That recently they sequenced the genome, they found the Omicron, and within 24 hours, they tell the health ministry, and within 48 hours, the parliament passed a law or bylaw to have a very tight regulations of the Omicron regulations. So these are the good success story that actually all started from the genome that can only be given by the NGS data. Okay, so this is another thing. Once you know the mutants or the variants, and then vaccine studies will be possible and very quick with the mRNA technology. It doesn't matter it's mRNA or recombinant peptides. All you need is to know the variants. If they have a breakthrough, vaccine breakthrough mutants, okay, so all you need to do is to crack the genome as quick as you can before they become an outbreak or pandemic again. So then try to design the vaccine. So these are the things that actually all of you now already know it. So the ACE2 receptor is the one that actually helps to bind to the SARS virus. And then the TMPRSS2, which acts as like a Caesar. So they cut it, the binding, and then they move the RNA into the human cells. From there, they hijack our genome system. So this is that simple, and published in Cell last year. So it takes two to a tango. That looks like, you know, the two parties are so close, where the protein-protein interaction, to be precise, okay? The human receptor and also the viral protein are so close that they actually can bind so strong, okay? So questions, okay? Now, COVID-19 are causing so broad spectrum of symptoms, actually from asymptomatic to very serious symptoms until to mortality, okay? So this is a wide spectrum of symptoms. And why is that the case? Again, yes, NGS are able to tell you the story, okay? It takes two to a tango. What I mean is that I have proposing a lot to use the NGS to sequence the viral genome, and that will also detect all the possible mutants, okay? That is only one, it's not two yet. The other one that take two to the tango is the human host. We responded in such a, you know, from symptomatic to asymptomatic and everything in between. The human host factor must play a role, okay? So these are the one aspect, or one to the tango, one party, which is the SARS-CoV-2 genomes. If you do a later search on 2021 genomes, okay, this data will get more and more complex, okay? And well, interestingly, this is the bat SARS-CoV-2 virus that isolated from the bat in Yunnan at that time, which is quite distantly related. But I think Lim Fa Wang in NUS recently have done a search on the bat in Laos and the upper region of Peninsular Malaysia. So they found those viruses are quite abundant in those bats that live in the wilderness, okay? And their genomes are quite similar to the one that we have. And I think that should be able to seal the debates between the lab leak and the what we call evolutions. The virus is actually zoonotic in nature, okay? But I don't want to get into that. Now, then this is the phylogenomics, that these three are only possible in 2021 that published in Nature Reviews Microbiology. Without the NGS, you can only resort to Sanger. If you remember the slide in 1977 that I told you, 400 to 800 base pair, even you outsource it, it is about 1,000 base pair. It will take a hell of a long time to get these three done. But with a panel sequencing or resequencing or de novo sequencing in the NGS, this a couple of months work can be actually just churned out in a matter of days, if not hours, okay? So that is actually possible. And the second one to the tango is actually human genetic variations that I personally believe, well, I'm not a clinician, particularly immunologist, but my gut feeling is that how come one patient responds so violently while the other are not, assuming that is the same variants that has been infecting the populations. So the human host genetic factor must play a role, or at least play some roles that contribute to the pathogenicity of this virus. So I think UK actually have started to sequence 100,000 genomes, and they are shortlisting the candidate which has been suffering from the COVID, from symptomatic to asymptomatic. I think they want to study that. I think this is something that our country should do as well. Two aspects, from the viral genomes as well as from the human genome as well. Okay, so, and these are some of the findings that has been reported. So the human leukocyte antigens does play a role in the disease outcome. Gene variants in ACE2, of course, these are the receptors that will directly bind to the virus. Innate immunity factors, ABO blood groups, and other unknown genetic polymorphisms. These are the things that we actually need to confirm. Okay, so, and I just want to share with you, although it is not part of the bacteria, doesn't fit into the studies of this today's seminar, but we get a Nature paper by this consortium work we get across the Asia, and then Malaysia contributed the samples, the human, mostly orang asli. So a quick genome analysis across all these about 1,700 human genomes, we actually found that, for example, warfarin are actually Japanese and Southeast Asian are very sensitive to these blood thinning drugs. So, and the BRCA1, BRCA2 mutations. So the Korea, Philippines, and Pakistan, particularly the India, has got a lot of different mutations. So what I'm trying to tell you is that if you do a pan-genome studies or GWAS studies, that is related to COVID-19, we will be able to pinpoint at the Asian context why certain patients are responding so violently, and while the other, we used to say children don't respond that violent, which I think is now wrong. Our youngest record in Malaysia is 3 years old, if I'm not mistaken. But we need to isolate those human cohort in order to study these genetic factors from the genome point of view, just like my Nature paper has shown. So, and that will be more or less to the last topic, bacterial methylome, that I would like to share with you. Is that all NGS can do for the microbiologist? Well, no, not yet. Okay. Now, so bacterial methylome is something that, in short, modifications in the DNA, particularly the methyl group being added into the C or the A, very rarely the G. Okay. So previously, this field of science in microbiology are almost negligible or ignorable, worse still. Okay. Why is that the case? To detect this methylated group in the 5MC, okay, you need a mass spectrometry. Yes, mass spec the nucleotides one by one. And you must have a very good spectroscopist in order to decipher the methyl group has been added into the DNA. Do it one by one. Well, even the smallest genome like Mycoplasma is about 0.6 megabases. You have to do how many rounds? 600,000 times of this mass spec, with the hope that you will find the methylations in the mass spectrometry. So that will be, seems to be, finding a needle in the haystack. That doesn't sound to be possible, okay? And for those of you who are not very familiar with DNA methylations in bacteria, so this consists of the RM system mainly, restriction modification system, where one of them is the methylases and the other one is the restriction or the endonucleases, okay? And they normally act in group, for example, the type 1, okay? And type 2 is the most and the simple one, most ubiquitous and the simplest one. Okay, this is a classic, what we call methylations. So in short, if the DNA is methylated, restriction enzyme will not be able to digest them. So this is the classical view of the methylations. They are type 3. So up to here, we only know the 3 types exist in the world. So, well, that may not be the case. NGS will be able to tell you, and I will be able to show you why and how, okay? With the availability of third generation sequencer, particularly the PacBio, I've been told the Oxford Nanopore has been able to do the same job, but our lab has been using the PacBio to do it. So this is the bacteria that we found that is named after me, my surname. And this is a new genus that has been found. And this is the Nobel Prize winner collaboration with Sir Richard Robert. So our group actually found these 2 methylases that actually based on the NGS in PacBio. Because PacBio functions by polymerase and they are sequencing by polymerase synthesis. So the polymerase can technically read the sequences from the template DNA. As they read, they have a certain kinetics, which is 1 minute, about 1,000 nucleotides being synthesized in the nascent DNA. But when the DNA or the template DNA has got methylation, the kinetics of polymerization of the polymerase will slow down. On this nanosecond or millisecond slowing down, we will be able to pick up that this region looks like methylated. Then we can actually use bioinformatic tools to identify that region and then followed by experiment to confirm it. So by this approach, we actually found these 2. And the red font is actually the novel discovery that we found these recognition sequences by the methylase that have not been seen before. And they are only present in the Chania multitudensis that has been reported by our lab. And this is a very nice paper that we co-authored with the Nobel Prize winner, Sir Richard Roberts. We are very good friends now. We worked for many years together. He came to our lab several times in Malaysia, and we have the honor to name our lab after him with his consent, written consent. Okay, with this, I come to the end. Thank you for your attention. But before that, I have to thank the amount of this work cannot be a work alone, no way. So these are all the collaborators that I have, my PhD supervisor in Nottingham, the French embassy that gave me more than 10 years of fellowships that allows me to work in Paris for more than 10 years while enjoying their wine and cheese and of course their scientific achievements, which is very valuable. This is a UK PMI 2 award that we have, my former supervisor, okay? And these are the Nobel Prize winner that is advising the HIR. And this is the one that worked with us, Sir Richard Roberts. And Rich is still working with me. And this is from the Harvard group that are advising us and work together on the microbiome. And these are the students, the creme de la creme, that enable all these publications possible. Thank you very much for your attention.
Okay, thank you very much, Dr. Chan, okay, for the very excellent and insightful as well as the very informative presentation. Okay, so we have now come to the Q&A session actually. So participant, if you have any question for Dr. Chan, please feel free to type it in the chat box. Okay, and I will read out the question for Dr. Chan. Okay. Okay, so perhaps I will just get the ball rolling, okay, by asking a few questions.
Sure.
Okay, Dr. Chan. Yeah, thank you for it's a really very informative presentation that you have given to us. Okay, so perhaps I would like to ask you, what actually would be the most challenging aspect, okay, when it comes to do this kind of study, you know, your microbiome as well as the metagenomic studies?
Dr. Chu, thanks for the questions. Excellent. I really have to think about it first. It depends. For example, if you work with environmental samples, my samples actually range from tropical seawater all the way to Antarctica soil. So you can imagine Antarctica soil, that will be very challenging. The soil samples is very precious, and it took 40 days to transfer on cold ice back to my lab, and that is actually quite a long years of work. But for example, getting the water to do the coliform in Butterworth that I mentioned, that is quite enjoyable. We even enjoy the black tone durian on the way to Penang, and that actually not challenging sample wise. So it depends on where it comes from. And I have worked with Prof. Yao before on the obesity patients. So getting two things. One is the adipose tissue, that is human subjects. That is very tough to get. We need to work with the surgeons. The other one is the bariatric surgery, and that is also very tough to get, and we very few. We only get one samples, and that also explains why most of my work I didn't deal with the human subjects. Those are very challenging. First, you need to consent, and I don't have patience to fill up the forms. I really adore the clinicians to fill up a stack of paper just to get consent and things like this. This to me is challenging. Of course, the other one is consistently challenging. Those times is the finance. One NGS used to take me 40,000. I used to tell the students, before you press start on my Illumina, think of your Toyota Camry. If you want a high throughput, you press start, you bomb the Toyota Camry. You want a low throughput sequencing, you press start, you lost a Toyota Vios. All my students know this. That's why we have zero error rate in my lab. I've been, putting psychological stress to them. These are the two challenging parts that I will see. Sample prep, all those things are quite straightforward. You just need to follow the protocols.
Okay.
Thank you, Dr. Chu.
Okay, thanks. How about, do you have any future projects that perhaps that you wish to do some unique environment?
Yes, I have presented it somewhere else, perhaps with Utah in Kamba campus, which is the Nepenthes, the Periocara. The Chinese call it Zhulongchou. And the botanists used to call the pitcher cup the digestive organ for the plant, and they have been stealing the glory of the microbiome for too long. The plant is not the only contributor. We believe the microbiome is actually doing a lot of work, and we have done that. That actually a collaboration with the Harvard group and the CNRS and the Natural History Museum in Paris. We use the mass spec to confirm the turnaround of the insect remains in the pitcher cup, as well as we use the NGS to confirm the microbiota, and we track the changes over the degradation of the insect remains in the cup. From when they are very young, when they are fresh, no insect, then we artificially introduce the insect into the cup. We monitor spatial temporal changes. So we actually confirm that the plant is not the only one doing the job. But unfortunately, some of you have seen the data before, and I try not to repeat the presentation. So I give you another presentation of this. But we are working on that Nepenthes. We're still continuing working on it.
Okay, thank you for your answer. Okay, the other question, what about the participant? Do you have any question for Dr. Chan? Okay, perhaps another question from me then. Okay, Dr. Chan, I've seen that you actually deal with a lot of different kind of genomes from samples, even from viruses and things like that. So basically, what about the approach? Let's say, for example, if you were to do any viral metagenome as compared to bacterial genome, are the approach the same?
Yes, but the downstream will be very different because microbiota, if you are talking about bacterial diversity, they have the 16S as a signature genes or RPOS genes, depends on your preference. Although 16S is always welcome, V3, V4 region, but viruses do not have a signature genes. So you need to have a very good virologist on board, that's for sure. You need to have someone who are very good at bioinformatics and who are aware of the virome databases. So that, and also virus is even more tricky. You have RNA viruses, you have DNA viruses. Some more positive strand, negative strand, and to me these are very cherevate organism. So downstream classifications will be very tricky, but it's doable. It's doable. You just need to look at, for example, if you are looking for the SARS-CoV-2's virome from the wastewater. For example, you just look at the spike proteins, which are characteristics to them. Then from there, you fish out. Because, and the plus point for virome is that most of the viruses, except megaviruses, most of the viruses genomes are within kilobases. So, and with that kilobases, it is really not tricky to handle them. And also the computing power, we used to have supercomputer in our lab. And we have about 20 units of them. It is very taxing in terms of finance to maintain that. But for virome, you don't need that. So there are plus and minus points in terms of, we call it the virome. And this is something, again, Dr. Chu, for your first questions, the virome has been in my heart for quite some times, not because of the pandemic. I have done the microbiota. I even have done the fungiome, the fungus diversity. The only thing left behind that I have not touched is the virome. And I know you have some very good virologists in Utah. I've been waiting for them to work together. Thank you for the questions.
Okay, I think there's one question from
Prof. Niao, I think.
Prof. Niao, okay. So Prof. Niao wants to know which human microbiome is still worth investigating.
Prof. Niao, thank you for very interesting questions, but very challenging. Of course, the hottest one currently, I used to talk to one Oxford professor who came to my lab. He's an expert in microbiome, and I told him, oh, so you're working in microbiome as well. Then he told me, who isn't? Even the ingrown nail, we blame it to the microbiome. It is very typical British sarcastic way of answering microbiome. But I think, Prof. Niao, the question is that the current one is actually, but someone has already done it, I know, the COVID-19 patients' microbiome. Because the ACE2 are highly expressed in the intestine, so that could be something that is worth doing. That's number one. Number two, I think now microbiome alone, to quote in Bahasa, very susah cari makan. So you may need to approach it from multi-omics point of view. And in that case, I would suggest what we call metabolomics. The presence of the microbacteria is there. Okay, so you see a very distinctive pattern between the healthy and the patient cohort. So just the presence itself is not equal to the disease onset, isn't it? There must be some metabolites that is actually secreting by the microbiome or groups of them that allows them to modulate the host response for that matter. Okay, so I think microbiome should be coupled with metabolomics that use the what we call mass spectrometry to do it. And that's why I always like to share with you the Weaver N story. So knowing the diversity, you only know a big data in the computer. I used to tease this to my bioinformatician teams. And after that, he left my lab and we are forced to become bioinformaticians. He used to say, the computer can digest this. I say, sorry, the bacteria can digest this. I say, how do you know, Dr. Who and who? He said, because I found the gene in the computer. I say, then the computer can digest the genes, not my bacteria. You haven't grown the bacteria in the lab yet. So I am saying that you need to grow the bacteria in order to verify that. I should have shared the data that we did an oral microbiome. we found the Elizabethingia meningoseptica. This is the one that causes meningitis in the human brain. And we are able to grow them in pure culture. They say that when your oral hygiene is bad, you run the risk of carditis, myocarditis, and also meningitis. So we found the pathogen. We grow them. So we are lucky in that case. If you cannot grow them, like what the VivoN data shows, then the other thing is that you need to look at the whole metabolomics and find out which metabolites are actually able to modulate the host response. We actually have a secretome data. Unfortunately, I think my slide is too long. I deleted it. I call it secretome. So I studied the environmental and multi-drug resistant Acinetobacter, one of the key AMR pathogens in the world, WHO classified. So we look at the secretions, and the environmental strain and the pathogens, the way they secrete their metabolites are different. So that can actually work as a biomarker or a way to modulate the host. We believe that. So we have the data in hand.
Okay, thank you, Dr. Chan, for the answer. There's one question from Hui Shan Leong. In your point of view, will the NGS replace the current identification method, such as the MALDI-TOF, in the future of clinical microbiology lab?
My gut feeling is no, because I really love the MALDI-TOF. 8 seconds, one bacterial ID. Even with the 16S, you take few days. Even you have the capillary gel electrophoresis, you have your own sequencer in your lab. It will take more than hours. I'm very sure. I done this all by myself when I was a student. Okay, I did my 16S. I did the sequencing of the 16S. It will take 3 days, 2 nights. I start work at 5 a.m. and finish at 8 p.m. every day for 3 days just to get the 16S. No way you can compete with, for example, the Bruker Daltonic MALDI-TOF mass spec. 8 seconds. I really love the machines. It won't replace. And that will be very good for clinical microbiology setting. I strongly urge your faculty to buy this unit. Then I will come and join you to do research.
Okay,
thanks, Shan Liang. Hui Shan, thanks.
Thank you, Dr. Chan, for the answer. Okay. Okay, so any more final question for Dr. Chan? Okay, so if not, thank you, Dr. Chan Kok Gan again, okay, for answering all the questions, as well as your very, very insightful and interesting presentation. Okay, so it is our pleasure to have you here with us today.
Thank you, Dr. Chu. Thank you, everyone.
Thank you, Dr. Chan.
Stay safe.
So with that, I shall pass on the mic to Dr. Alicia Ho. Dr. Alicia.
Thank you, Dr. Chu, and also Prof. Chan. So now I would like to present the Certificate of Appreciation to Associate Professor Dr. Chan Kok Gan for his contribution as a speaker in this webinar. Prof. Chan and Dr. Chu, please keep your webcams turned on as we capture a photo, yeah?
Oh, thank you.
So, Dr. Wong, whenever you're ready, please let us know, yeah? Okay, so I guess
one, two, smile. Okay, so let me see. Okay, so can we get the second photo? So 1, 2, smile. Okay, thank you.
Thank you.
Thank you, Dr. Wong, and thank you again, Prof. Chan, for your astounding presentation. So
Thanks.
now, I would like to present the Certificate of Appreciation to Dr. Chu Kok Cheong for his contribution as a moderator. Dr. Chu, you have your webcam turned on, right?
Yes, yes.
Yes. Okay, Dr. Wong, again.
Okay, 1, 2, smile. Yeah. Okay, one more. 1, 2, smile. Okay, thank you.
Okay, thank you very much.
And thank you again, Dr. Chu. All right, let's move on. Now for our second talk, let me do the honor of introducing our final speaker for the day, Dr. Joshua Ang Sinter. So as you may have read, from our event flyer, Dr. Joshua Ang graduated with a BSc Honors in Biotechnology from Utah, and he subsequently obtained his PhD in Medical Entomology from UNIMAS, Sarawak. So during his PhD, he worked with Dr. Khatijah Yaman and also the famous Professor Balbir Singh to incriminate previously undiscovered Anapholic vectors of the zoonotic malaria parasite, which is endemic in most Southeast Asia, the infamous Plasmodium knowlesi. His research interest involves the usage of advanced molecular methods in controlling vector-borne diseases, especially malaria and dengue. He was the holder of the Early Career Scholar Grant Program and won an award in the International Congress for Tropical Medicine and Malaria in the year 2016. So 3 years ago, which was 2019, he started in the Professor Luke Elfie's group and Fulbright Institute UK to focus on control of mosquito via genetic manipulation. Let us now welcome Dr. Joshua Ang. Dr. Joshua, over to you.
Hi, can you guys hear me?
Yep, let me stop the
Okay.
sharing and let me share the screen, yeah?
Sure, thanks. Okay. Can you confirm that you can see my slide?
Yep, I can see your slide. Clear.
Okay, thanks. Thanks for the introduction. I guess first of all, I'd like to thank the organizers for inviting me for this talk. It's an honor to be back. It's good to be back actually, virtually, although not in person. So I'm going to start the talk. I will dive right in. And then if you have any questions at the end, feel free to ask any questions. Today, I'm going to talk about an overview of the development of such system, Cas9-based gene-drive systems in mosquitoes, without going into too much of the nitty-gritty, because I appreciate that most of us after this talk won't actually be working with mosquitoes. But I hope that you'll get something from the aspect of, say, Cas9-based genome editing, or technical nuggets that you could. Let me just check that you can see my laser pointer. Sorry, Dr. Alicia, are you seeing my laser pointer?
Yes.
Yeah, I can see the
Okay.
laser pointer, yeah.
Good. Okay, all right. Let me just first start by introducing a few major diseases, major infectious diseases in the world. So we have malaria, which is endemic in a lot of countries in blue here, specifically Falciparum. That's the main killer of malaria parasite in the world. WHO estimates that there are 229 million cases, 409,000 deaths in 2019, and the majority of them were actually children under 5 years. And as Ms. Alicia was saying just now, I used to work with Plasmodium knowlesi, which is the predominant malaria parasite in Malaysia right now. And as you can see here, malaria cases in total is actually reducing year by year, generally. But what you see increasing every year is these bars that are in green color, and they are zoonotic malaria. And this is what I was working with previously, because we're seeing increasing cases of Plasmodium knowlesi, the zoonotic malaria parasite cases in Malaysia. The next one is actually dengue virus. We are pretty familiar with dengue viruses in Malaysia because we live in Malaysia, and it's estimated that each year there could be 100 to 400 million infections. The next one would be Zika virus. We're also pretty familiar with the outbreak several years ago. And actually, the main or the common denominator of all these diseases and other diseases is actually the mosquito. These diseases that I've just mentioned just now are actually mosquito-borne diseases, and they're all transmitted by mosquitoes. But just a fun fact, not all mosquitoes transmit diseases. And to put things into perspective, there are thousands of mosquito species, but only a handful of them actually transmit diseases such as these. So the main conventional approach in controlling mosquitoes in the wild is actually by insecticides. We living in Malaysia are very familiar with mass spraying of insecticides when there's a dengue case. But although insecticides do kill mosquitoes, mosquitoes all over the world have now developed resistance to the main insecticides that we use. And one more disadvantage of using insecticides are actually the environmental harm that they pose to the environment. So insecticides do not only kill mosquitoes that we care about, they also kill other insects that might be beneficial to the environment, and sometimes even aquatic organisms. So this has actually encouraged the development of genetic control technologies, which are species specific, because the genetic control methods require mating of modified mosquitoes to wild-type mosquitoes. And in genetic control, there are two main goals that we aim to achieve. We have this population suppression approach. We're not going to talk about Wolbachia, but Wolbachia is one of the ways. We're just not going to talk about it today. We're mainly going to talk about gene drive. So in a population suppression approach, what we aim to do is to spread a gene into the wild population, which will over time reduce the population of the mosquitoes. And therefore, with less mosquitoes, you have less infective mosquitoes. With less infective mosquitoes, you have less infectious diseases. The other method is actually to modify them. So population modification approach, where we introduce a gene that is beneficial to us. In other words, these genes could be anti-malarial genes, antiviruses gene. So when we modify the population of the mosquitoes in the wild, the mosquitoes will no longer be able to spread the diseases. Again, these methods are species specific and they affect only the species that we care about, and they don't kill or modify the other species, other insects in the wild. Now, so let me digress a little bit about to talk about homing endonuclease genes, and then you'll find out why later. So there are these homing endonuclease genes called HEG. Thereafter, I'm going to mention it as HEG for notational purpose. HEGs are found in nature, and basically what they do is that they embed themselves either in introns or inteins. Introns is just mRNA being spliced. Intein is just after translation, the protein is spliced into proper proteins. So they embed themselves into either exon or proteins, and eventually they will act as nucleases that cleaves the genome at the homologous chromosome and then inserts themselves into the homologous chromosomes. So these are like selfish genetic elements. They cleave the wild type chromosome, embed themselves in the wild type chromosome. So an organism that has actually only one copy of the HEG would then be able to spread, will be homozygotes essentially. They will just copy themselves and then increase their frequency in the population. Now then, but the problem with this is that the recognition sequence of this HEG are usually really long and very, very specific. It is very difficult to program these HEGs for use in actual research. Then come about 10 years ago, the discovery of the use of Cas9 and guide RNA to just by using like 20 base pairs and GG at the end, 23 base pairs of sequence. And you can actually specifically target almost anywhere in the genome. This discoveries has dramatically improved the flexibility that scientists have to carry out genome editing work. And for those who are not familiar with Cas9, I'm expecting most of us are quite familiar, but I'm gonna try to explain how this works anyway. So you could express a Cas9 and the guide RNA from a plasmid And the guide RNA will then guide the Cas9 into a genome, into a specific locus in the genome, and it will then create a double-stranded break on that specific site on the genome. Now, you could do two things after that. You could allow the cell to repair it by non-homologous end joining, or NHEJ, in an error-prone way. So it would just repair because it needs to repair. The cell wouldn't allow, in most cases, the chromosome to just be broken. It will just die. So it will have to repair. And then if you cleave a gene, an exon, or a coding region of a gene, the gene would usually be disrupted due to out-of-frame mutation, or sometimes in-frame mutation might actually disrupt the gene as well. Now, you can do another thing. You could provide the cell with a repair template. So you kind of trick the cell to think that, oh, okay, this is where I should repair it from. So you have to have homology arms, which are colored in green and yellow here. So these actually corresponds to this part of the cell, and these actually corresponds to this part of the cell, of the chromosome. So if you cut a chromosome, you provide it with a repair template, it will at some frequency repair the broken gene with your repair template. Now then scientists started thinking, can we then program Cas9 and guide RNA to turn it into a HEC? So basically, if you could put a Cas9 and guide RNA into the chromosome, then you could potentially make a gene drive. How do we do it then? If you insert a Cas9 and guide RNA into this locus, and this locus, this endonuclease targets the homologous chromosome at the exact same location, then at some frequency, when it creates a double-stranded break, it will copy the homologous chromosome, right, and make this heterozygote individual into a homozygote. And if this occurs every generation, you will then eventually get a whole mosquito population that has your gene, that has your Cas9. And of course, you can stick a cargo, a gene of interest, into with your endonuclease gene. So every time this gene gets copied over, you have your cargo with you. But of course, at some rate, if homologous recombination or homology-directed repair does not occur, then you will end up with NHEJ again, where the drive allele, this endonuclease gene, is not copied, and the wild type gene gets cut and repaired in an error-prone way. So I'm going to talk about two of the earlier studies of HEC drive systems in Anopheles mosquitoes. I'm going to provide one example for population modification and one example for population suppression in this talk. So the very first one was done in Anopheles stephensii, an Asian malaria vector. So what they did was they targeted the KH gene. It's a gene that codes for eye pigmentation, and it's recessive. So if it's haplosufficient. So if you have one copy of this gene that is working, the mosquito will have a black eye or a normal wild type eye. But only when both copies are knocked out, the mosquito will present a white eye phenotype. So they insert a Cas9. This is just a promoter, Cas9, and guide RNA. This is one part of the drive allele or transgene. And this is the second part. This is just a marker to tell us that this has actually inserted into this region. And these are anti-malarial antibodies. So this is population modification. They disrupt the KH gene, inserts into it, and they expect that the KH gene, when disrupted, should not have a fitness cost because you want to modify the population. You want it to be as fit as the wild type. So when they have generated this mosquito with Cas9, guide RNA, marker, antibody, they found the marker to be present, very faint here, but they'll show up in the next few slides. So you see This is the eye. This is a wild type normal eye of a adult mosquito. This is a normal eye of a larva, mosquito larva. And in the wild type, you don't see the fluorescence, but in the genetically modified mosquito, you see the fluorescence in red here. So under normal Mendelian inheritance, when you cross a heterozygote drive, the mosquito in red, to a wild type mosquito, under normal Mendelian inheritance, you will have 50% of your progeny inheriting the DS red, the marker, and the drive allele, and 50% of your progeny would actually be wild type, right? So, but if you have a hack drive that homes 100%, so that copying over, after cutting, copying over, that phenomenon, we call it homing. So if homing occurs 100% of the time and cutting occurs 100% of the time, you would then be able to skew the inheritance of the drive to 100% of the time. So if it works 100% of the time. So under an ideal world, in an ideal world, all progenies would have dark eye, again, because the KH allele is not disrupted. Dark-eyed, but inheriting the drive allele, and then none of them will be wild type. All right, but what they found was a panel of different phenotypes. So when they cross a drive heterozygote to wild type, they found wild type phenotype. They found those that did not inherit the drive but were white eyes, and they found, interestingly, some mosaics. So basically, in these mosquitoes, some of the cells still had KH alleles that are working, at least one copy, but some of the cells had no copies of functional KH alleles. And it differs depending on where they got the drive from. So in the case where they crossed the drive male in red to a wild type female, they saw what we would expect from a super Mendelian inheritance, where 99.6% of their progeny got the DS rate, or in other words, the drive allele, but are dark-eyed. Only 0.4% did not have the drive allele and were dark-eyed. So this is fine. This is pretty normal, something that's expected. But when the drive allele came from a mother, a female drive heterozygote crossed to a male, again, they still saw an increase of inheritance in the drive allele, shown by DS red inheritance. So these two are DS red positive. So they have the transgene, 85.9 and 13.3%. That's 92%. Sorry, 99.2% of the time they got the transgene. But most of them are white eye or mosaic, unlike in here where everything was dark eyed. The hypothesis was that in these cases, the mother, the drive mother, actually deposited Cas9 into their embryo, which then starts cleaving the KH allele that came from the father, if that makes sense. So therefore, most of them had mosaic or white eyes because the wild type allele that was paternally inherited got cut by deposited nuclease activity. Now, more interestingly, they took these, all right, these drive positive white eye mosquitoes and these drive positive dark eye mosquitoes and outcross them to wild type again to look for inheritance to see if homing continues to occur. And they found that, take note of the dark eye and white eye here. They found that the next generation, when they cross the dark eye to wild type drive allele, to wild type, homing still occurs. So you still have high inheritance of the transgene. But then when they cross the white-eyed drive inheriting mosquito to wild type, drive stopped occurring. This is almost like Mendelian inheritance. So the hypothesis, again, was that in a male transgenic, the male contributes the sperm, into the embryo or the zygote. The female contributes its chromosome, and then only in the germline or the germ cells of the male, cutting and homing occurred without disrupting the other somatic cells. And therefore, you have a dark-eyed progeny where its germline or germ cells were converted, but not the somatic cells. Therefore, you have supramendelian inheritance, but the father or the drive-carrying mosquito was still dark-eyed. But in the case where the transgene came from the mother basically deposited Cas9 and nucleus activity into the embryo, shown in blue here. And because of that, when the father's sperm fertilized the zygote, Cas9 immediately cuts, or very quickly cuts, the chromosome that came paternally. And that wasn't a right time for homing to occur. So instead of homing, NHEJ occurred, disrupted gene without homing the transgene. And therefore now you have an embryo that develops, but is now converted, but not converted the way we want, converted in an NHEJ way, not HDR or not homologous recombination. And therefore you get a wide-eye progeny and no homing, because basically by the time cutting occurs in a germline or wants to occur in a germline, it is not cleavable anymore because at the beginning cleavage has occurred. Now the second study is a suppression drive in Anopheles gambi. The target gene has to be slightly different here. And in this case, they were looking for a gene that is female sterile but recessive. In other words, they're looking for a gene where both copies, if the female has 2 functional copies, it will be fertile. If it has at least 1 functional copy, it will be fertile. But then if it loses both copies, the female will be infertile, but the males will still be fertile. So it affects only the females and only when both copies of the alleles are knocked out. So they were testing 3 genes by inserting just a marker, a GFP fluorescent protein, into the gene to disrupt it. And they found, so these 3 numbers are just 3 candidate genes. And they found that in heterozygote, so HDR GFP / positive is a heterozygote. If you see GFP, that's a homozygote. So a heterozygote knockout or knock-in, they produce normal number of larvae. But then if it's a homozygote, the larvae number reduces. Similarly, in this, the next gene and the next gene, they see the same thing. And these are only for females. They did not check males. And then the next step was, of course, to insert the Cas9 and guide RNA and a marker into the target gene to disrupt it, and hopefully it homes. Now, in this case, for population suppression, you don't really need a cargo. So in the previous case for population modification, there were 2 antibodies, anti-malarial antibodies. But in this, you don't need it because you just want to suppress or reduce mosquito population by disrupting the female fertility gene. But what's important here is the restriction of nucleus activity. Now, what you like to happen is to have a drive heterozygote, which is heterozygote in the somatic cells. All right. So in the somatic cells, it should be a het because you would like this female when heterozygote to be fertile. You don't want to knock out both copies and make the female infertile when it only has 1 disrupted copy. But what you want to happen is to have homing in the germ cells. So you really have to restrict nucleus activity to a small part of a really specific cell type in the mosquito. And so when homing occurs in a germline and you cross it out to wild-type mosquitoes, you would expect most, if not all, of the progeny to have at least 1 copy of the drive allele. And we hope if the nucleus activity is really restricted to the germ cells, then these mosquitoes, especially females, would be fertile. And then eventually, as this spreads, you will then get female homozygotes, which are not fertile. Female zygotes are still fertile. That's the ideal world, right? And as it spreads, you should be able to reduce the population because you will get more and more females which are infertile, and the population should just crash. But that's also not what they saw. But there are good news and bad news. So good news is that in all of them, they saw super Mendelian inheritance, where the inheritance of the red fluorescent protein, in other words, the transgene, was increased to about 90%. In some cases, near to 100%. So these are 3 genes. When they cross their drive heterozygote to wild type, instead of 50%, they found more than 87% of inheritance of the transgene. Now, what they didn't want but had occurred was that their heterozygote females were infertile. So in this case, the females had almost 0 eggs, 0 larvae. And in this target gene, there were still some eggs, but then the larvae number were pretty low. But in the males, you see a normal fertility, which is great. So leaky expression, or in other words, expression of nucleus activity at unintended location or time, basically made the heterozygote females infertile. And this would actually reduce the ability to spread in the population, and not ideal. Fast forward, long story short, 5 years later, cage studies have now been conducted with new versions of the population modification hack drive in Stephen's eye, the first one that I showed. Briefly, these are cage studies, and in each cage they had 200 mosquitoes. And these are the ratio of genetically modified mosquito to wild type mosquito that they put into each cage. These are the ratio of males actually. So they start the cage by 100 males and 100 female adults. And in this case, you see that this is the starting frequency of the drive transgene. This is the starting frequency when the ratio was 1 to 3. And this was the starting frequency when the ratio was 1 to 9. And they found in all 3 replicates and 3 different release ratios, the transgene was introduced and reached genotype fixation to a very high level, in other words, in a matter of like 12 to 13 generations. And these in green are actually mosquitoes that carry the anti-malarial antibody. Now, the largest cage study, as far as I'm aware, is this one, in a really huge cage, where they tried to demonstrate population suppression hack drive in Anopheles gambi, just another species of mosquito. And they tried to mimic natural habitat of the mosquitoes. They have the swarming arena, wet resting site, and etc. And of course, they have a dusk and dawn, light cycle, dark cycle, just to mimic a natural population. And they found that, so this is the name of the strain, AQQFS1 of the population suppression strain. They found that the egg output after, say, around 250 days in one replicate and around 325 days in the second replicate to drop to 0. So they started a cage with about 400 pupae or adults, and every 2 weeks they replenish 400 more adults from the same cage. So basically, if you start from a cage with 400, 2 weeks later you get whatever's inside, put 400 in more. And over time, egg output dropped. This is with 12.5% of introduction. So the introduction frequency was lower in this case, in A. And in B, introduction frequency was 25. And this was just a control. This was just wild type. And you see that the egg output in wild type cage is normal. It doesn't drop, it doesn't go up. And in this case, this measures, this follows the drive carrying mosquitoes that are in the cages. And over time, you see that by 250 days or 300 days, all of the mosquitoes in the population had the drive. In other words, the drive has spread in the population over about a year, less than a year actually. And this as well in a higher release frequency. So in short, I think the field of genetic control has developed quite a lot over the past 10 to 20 years. And they're now looking to work on other stuff in terms of regulations, in terms of making the drive better, and in terms of making a drive more localized. And I think I'll end here. And if you have any questions, I'll be more than happy to take questions. Thank you.
All right, thank you, Dr. Joshua. Hang on while I share the screen.
Sure.
And participants, anyone who are having any questions, burning questions to ask Dr. Joshua, please do so and type in the chat box, yeah? Or you can see my screen, right?
Yes.
Great. So let me see if there are any questions. Okay, no questions. Right, never mind. I'll do the honor of asking. So, Dr. Joshua, maybe you, I think you don't know me well, or this is your first time knowing me. I'm actually a researcher in mosquitoes. So my
Yes.
BSc, my master's, and even my PhD work is all about mosquitoes. So my master's was about Anapholic, and then my PhD was about Aedes. So, and your work is basically on the Anopheles mosquitoes, based on your right?
Our lab actually works with Aedes aegypti, Culex, and a few other insect species as well.
Right. So
yeah,
your work regarding the modification on the Anopheles mosquitoes, restricting their fertility, and then the other one is on actually adding a anti-malarial antibody in them, right? How is it different, or how is it similar to the one 10 years ago when they were releasing, you know, genetically modified Aedes aegypti mosquitoes in a setup? Yeah, how is it different or similar to the one you're doing now? Yes,
that's interesting. So I think the one that you're talking about is released by Oxitec, if I'm not wrong.
Yes.
Yeah. Okay. So my current boss, Luke Alfie, is actually the founder, one of the founder of Oxitec.
Okay.
And 10 years ago, the technology was called SIT. It is sterile insect technique. So those needs to be replenished. So they don't spread. You release them, and they are basically infertile males. So you release them, the males will mate with wild type females, making the females infertile because the sperms are infertile. So they have mated, but they're infertile, and they could not mate with other wild type females once they have, the wild type females could not mate with other wild type males once they have mated with the SIT males, if that makes sense. So the females would either produce no eggs or produce eggs that are not hatchable. And so when you release them, you will reduce the population for a bit, and then you have to release more, and then you have to release more.
So in your case, for your work, it's like you don't need to replenish. Once it's released, it's able to sustain itself.
That's right. That's right. So just to make it, just to clarify, the work that I've introduced just now were not directly done by us. It was it's an overview of what's happening in the field in general. So in that case, in the hack drive, yes, you do not need to replenish. It will just go on and on and on until resistance forms.
Okay.
Yeah.
Right. So from the review that you showed, it is mainly a lab setup, right? You're having cages of 400 over mosquitoes with the gene, and you're looking at its prevalence. So were there any sort of like small scale field trials conducted?
As far as I'm aware, not with hack drive. There are field trials being carried out at the moment with male sterile, very much like SIT.
Mm-hmm.
Yeah, but not with hack drive, I don't believe. Yeah, but I might be wrong. Yeah, that's as far as I'm aware.
All right. Oh, I see that there are some questions there. Great. Okay. Now a question from Anthony T. What is the anti-malarial genes inserted? What are the functions? Well, okay, there's a lot of questions here. Is that possible there is any malarial strain that resistant that may pose challenge for this model? Okay, there are 3 parts to this overall question. So
yes. So let me just clarify as well that our lab are not working with population modification approach. So again, that was published by another group. As far as I know, I'm going to answer it at the best of my ability. The antimalarial genes are single chain antibodies. This was published in a PNAS paper. And is it possible that there are any malaria strains that are resistant? Of course, resistance will appear over time, no matter what strategy you use, whether it's insecticide, whether it's genetic control. Natural selection will select for those that are more fit for every organism. So resistance will definitely occur. Even if it doesn't happen now, it will happen eventually. That is pretty certain. Yep.
Yeah, okay. I hope that answers your question, Anthony. And we have a question from Jackson. Will Cas9 technology be able to eliminate or at least reduce the spread of other disease carried by mosquitoes? What are the challenges when using Cas9 to work with mosquitoes?
Cas9? Sorry, let me just read it just to make sure I understand the question. Eliminate or at least reduce. The first question is yes. Yes, as was introduced in the slides just now, it will be able to reduce the spread. So if you could reduce mosquito population, it will eventually reduce disease spread. And if you could modify the population as well, you will reduce spread of the disease. What are the challenges when using Cas9 to work with mosquitoes? It is quite difficult because, well, in many aspects actually, because most of the time when you work with Cas9, you work with Cas9 in cells, right? You work in cell culture, or you express it in an in vitro environment. But once it goes in vivo, you have to think about where you want Cas9 to be expressed. That's a huge pain because you need the right promoter to express it in the right place. That's really a huge pain. 10 years ago, people had to look for the right promoter to get Cas9 expression. And then the promoter strength, if Cas9 is expressed too strongly, you impose a fitness cost on the mosquito. If it's too, if the strength is not enough, you get nothing. So there's a fine balance when it comes to in vivo. I think that's one of the main challenges.
All right. Yep. I hope that answers your question, Jackson. Well, frankly, Joshua, I do admire your courage in doing this work, as I have never dealt with molecular work when it comes to mosquitoes. But I'm sure I can learn a lot from you. Okay, next question is from Yuan Teng Hui. Out of curiosity, does reducing the population of mosquitoes will indirectly affect the population of other mosquitoes? Oh, okay, this is something that I've actually wanted to ask. For example, reducing food source for organisms that eat mosquitoes. Okay, it's about ecosystem, right?
Yes. As far as we know, mosquitoes are not a niche species. In other words, once we wipe them out, if we can, we actually can't. It's near impossible to wipe out all mosquitoes actually. As far as we know, it's not a niche. And when mosquitoes are wiped out, birds, fish, bats that feed on mosquito adults and larvae, they actually could find other food sources. Now, if you were to wipe out mosquitoes, insecticides would actually be easier, I would think, because insecticides don't choose species. You could literally just spray insecticide and all mosquito species will die when it comes to the contact, if they're not resistant, of course. But with this method, it's arguably more environmental friendly because it targets only one species that you care about. Just going back to the challenges, I just thought about one more. Challenges when working with mosquitoes. It's the generation time. When we generate a transgenic strain, it takes about 2 months to make one by from injection of the embryo, rearing them, outcrossing them, screening. And then when you screen, you found that, oh, there was no transgenics. You spend another 2 months just to get there. And then after 2 months, you spend many, many more months to assess whether they work or not. And if they don't, you go back to ground zero, start injecting again. And it just takes ages.
Yeah, I totally agree with you when it comes to the stress of culturing mosquito. Yeah,
Yes.
for them to grow from the egg to the adult, it's a long process of waiting. And then you have to make sure that they are able to sustain in the environment that you're, breathing them in. Yeah.
Yep,
Okay.
that's right.
Another question from Anthony here. For
Mhm.
both modification and suppression models, have you tried out the intra-breed between Anapholic and probably others like our bitters? Okay, this is for you to answer, yeah.
Yes. So for Anopheles and Aedes, they are mosquitoes from different genera. I don't think it's possible that they will interbreed. But if you talk about closely related species, for example, Anopheles gambi, sensu stricto, strictly Anopheles gambi, and say Anopheles colici, we have never tried that. Like sibling, closely related species, put them in a cage, see if they interbreed. As far as I'm aware, well, we have never tried that ourselves. As far as I'm aware, I don't know if anyone has tried that actually. Yeah.
So would that be possible? I mean, this is my question to you. Would that be possible? Not talking about between the Egypti and Anopheles, but between closely related species of the Anopheles. Like,
so if they interbreed, what would happen? I guess that's the next question, right? What would happen? It would have to depend on the target site. So if Cas9 and guide RNA could target the locus in that species, in the unintended species, let's say, it could potentially cut and home. But target sites, because they are sequences, they're DNA sequences for proteins, right? And even in different species, proteins have different DNA sequences. So it might not be, it might be difficult actually to target a different target site, well, to target the same gene in a different species, if that makes sense. It would be difficult, but we have never tried it.
Yeah, okay. Next question from Paul Davis. Hi, Dr. Joshua, nice talk. What about the lifespan of this genetically modified mosquito? Any effect on this? How would their breeding and feeding behaviors?
That's interesting. So it depends. It depends if you want to modify or suppress. So of course, because in a suppression drive, you target a fertility gene, right? In most cases. So even the heterozygote with one disrupted copy of the fertility gene, they would be expected to work less fitly, or they are less fit compared to wild-type mosquitoes. And of course, the homozygote females are not fit at all. They're sterile. That's what we want. But in modification strategy, in the lab, they are, as far as I know, as fit as wild-type in the lab. It remains to be seen what happens when they're actually released. But I think take many more years. Yeah.
I'm sure when it comes to gene modification, it takes many years to perfect it.
Yes.
So let me
Yep.
see if there are any more questions. Oh, okay. Anthony is still asking one more question here. So if this model works, how would you see yourself in a future world where we are surrounded by all the mosquitoes with different anti-parasite and antiviral genes?
Yeah, that's interesting. I think first of all, the WHO has recently published guidelines of how we should proceed when it comes to genetically modified organisms. Not just with mosquitoes, actually, because genetic modification is very, very prominent these days because of Cas9. And so I think researchers will have to at some point decide where they want to release if it actually happens and how they want to do it. They have to sit on a table and discuss. And that's why we are more, we lean towards population suppression, because then if you suppress them, they just die. They don't stay around. But of course, you could have some communities where they would prefer not wiping out the mosquitoes. I think there are a few things that needs to occur. Communication with the community, where you want to release it. There needs to be a committee of how this is going to work. And yeah, I think that will have to be decided by people, not just me.
Okay. All right. I hope that answers your question, Anthony. Yeah. So regarding these challenges, yeah, when it comes to genetically modified mosquitoes, have you encountered or have your team encountered any challenges when it comes to the bioethical areas?
But sorry, in what area? Sorry.
The ethical implications regarding these genetically modified mosquitoes. Did your team have any issues?
Not us personally, because we don't deal with the lawmakers, right? We work under a grant that was written by the boss, right?
Oops.
So most of the time, having discussions and talking. If that answers your questions.
Oops, I think I lost you halfway at the end, yeah, Joshua. I think you connected.
I was just saying we don't personally deal, we don't personally involve ourselves with like ethical issues, because we work in a lab, and usually it's the principal investigator who goes around talking with people, having discussions. Yeah.
Right. So you let your boss deal with all the lawmakers, yeah?
Yes.
All right. Okay. There is one question here from your, should I say ex-boss? Eddie, how stable are the gene edits by the Cas9 approach with regard to your application? Is this approach being studied on other vectors such as those of Lyme disease, sleeping sickness, or those of livestock importance?
Yes. Let's see. The first question: how stable? I think so. It's pretty stable as far as so once we integrate the transgene into the genome, it's pretty stable. It doesn't just get cut out of a sudden. It stays with the mosquito, or it stays in that chromosome for a while, or it homes when it cuts the wild type chromosome. How stable it is in the population would have to depend on the fitness cost, right? If it's fit, the mosquito with that transgene will stay in the population for a while because it's fit, right? But if it confers a fitness cost to the mosquito, then in the population, selection will select against that mosquito with less fitness cost. Sorry, that mosquito with less fitness, right? So over time, it will just drop out from the population. That's the first part of the question. Second part, yes, this approach is being studied. In other organisms, one that I know of is actually invasive organisms, invasive species, such as mice that invade into some islands and cause devastating environmental impact on the island habitat or the island vegetation. So this is being pursued in other organisms for sure.
Okay, so thanks for your answer, Eddie. I hope that answers the question.
Thank you.
Oh, there's one more here, just popped up. Chen Yang, this is your question. The two methods mentioned is by reducing the fertility of the host vector, while another is by increasing the endogenous resistance of the vector. So based on your experience and on literature, which approach is more feasible or popular?
Oh, there are two parts of the question, feasible and popular. I think in terms of feasibility, population modification is easier because you don't really care if the Cas9 is restricted in the germline or not, right? Because you could cut everywhere in the body and the mosquito will still be fit and it could still carry on the transgene. It could still carry and spread. But in terms of popularity, people I've spoken to seems to lean towards just getting rid of them, not replacing them with mosquitoes.
I totally
with other mosquitoes, yeah. And sorry.
I mean, I totally understand that. Yeah, people just want to get rid of them. Like, don't even want to have one mosquito flying around you.
Yeah, that's it exactly. And with the scientists that I've spoken to or heard from, especially from sub-Saharan Africa, they just want to stop malaria really. And it's taking time. The regulatory process is taking time, and every year so many people are dying. And well, good news is that recently the WHO has approved the use of the new vaccine for malaria. So that's good. And I think the field is just hoping that something could just stop malaria.
Yeah. So because malaria is more of an issue in the African countries, not so much in Malaysia or in Southeast Asia. So we don't really feel the impact of malaria here. More towards the dengue virus will be the main issue for Southeast Asia. Okay, so let's see. Any more questions? So no more questions, but my last question, I think this will be my last question. What did you choose, or why did the group choose?
Connection.
Hello? Joshua, can you hear me? Hello, Joshua?
Yes, I hear you now.
Yep. So
yes, can you hear me now? Yeah.
Hello? Joshua?
Yes.
Okay, I can't see you. Oops, I think Joshua is no longer in the group. Let's give some time. While we wait for Joshua to come back in. Let me see. Oops. Joshua, are you there?
Hello, can you hear me now?
Yeah, you're back. Okay, finally.
I'm sorry.
You disappeared for a while there.
Okay. Sorry, where was I?
No, I haven't asked my question yet.
Okay.
I was saying that this will be my last question.
Okay.
Regarding the choice of Anapholic. One was Anapholic stephensii and the other was Gambi, right? Why these two among of all the Anapholics?
Oh, yes. So these are the primary vectors that actually meant a lot for people most affected by malaria. So Gambi is the main primary vector for sub-Saharan Africa vector for malaria. And stephensii is just really bad news because stephensii is an urban malaria vector, while Gambi is not. But in recent years, stephensii has slowly creeped from Middle East into Africa. And so if it goes unstoppable, it's going to create a huge problem in urban malaria in Africa, in sub-Saharan Africa. So something has to be done really quick, or we'll see the effects in years to come.
So your team is actually working more towards the Anopheles species, which are more of a problem in Africa, but not so much of in Southeast Asia, right?
We are working in Stephen's Eye at the moment, which is slowly creeping into Africa. And it depends on if we will eventually get this out or not. We don't know. But we're trying to develop something that could be used and people be happy to use and get it ready.
All right. Thanks so much, Joshua. I mean,
Thank you.
I can really talk to you a lot about the mosquitoes, since here in Utah, I'm kind of like a sole or lone ranger when it comes to mosquito research. Thank you so much for your time, and let's proceed.
No, thank you.
Thank you. Let's proceed now to the next one, which is, of course, I need to thank you, right? So here is to present the Certificate of Appreciation for your contribution as a speaker in this webinar. And okay, your webcam is on. So let's have a picture. Dr. Wong, whenever you're ready.
Okay, 3, 2, 1, smile. Okay, one more. We are. Okay, another one. 1, 2, 3, smile. Okay, yes. Thank you.
Great. It was nice listening to your work, yeah, and the team that you are at, Professor Luke Alfie's team, yeah. Okay, great. Let's move on. So let me wrap up. Once again, I would like to thank our two speakers, Associate Professor Dr. Chan Kogan and Dr. Joshua Angsinter, for their truly insightful sharing and imparting such invaluable knowledge in the advancements of molecular technologies, which we believe will bring about great breakthroughs in overcoming infectious diseases. We look forward to hearing more new discoveries from them. Also, we would like to thank Dr. Chu Kwok Cheong for being impressively well prepared and extremely professional in the way he moderated the first session. We are truly grateful for the excellent job. So participants,